Okay, on this one is chapter 20. I don't even know what chapter I had, 20, I guess. We're going to talk about some of the DNA tools, and then we'll break it up also into some of the other things that we some of the big name things you might have heard of, I guess. So let's talk about DNA tools first, and some of the sequencing. So the whole idea with DNA sequencing is trying to figure out, what is the different nucleotides, what's the order. Okay, and so there's these big, huge processes of basically taking the genome. Genome is basically all your genes together, and breaking it up into fragments, and then trying to figure out ways of copying that, and then basically reading it. It's basically trying to get the code. Once you get the code, then figure out what it means. So we've done a lot of different techniques. DNA cloning is one, where genes are inserted into a plasmid, which is considered a cloning vector. So we're getting bacterium to do what we want it to do, and then we get copies of genes so we can get it to make stuff for us. We do this with human growth hormone to help, so we can get bacteria to make it for us. We're doing it in plants. We're doing it for bacteria waste and heart attack therapies to make proteins. So we're getting bacteria basically to make pieces of DNA for us is what we're doing. So we can grow it in culture, so we can grow it cheap. Insulin, we used to actually make in cows, and cows are big and cows are expensive to keep. And now we're actually, insulin is being made by bacteria, which is a lot easier to keep. So I have an idea. Recombinant DNA plasmids. Recombinant means just to kind of remix. And so we can get bacteria to do this in their plasmids. What we do is we do these restriction cuts. We use these nucleases to cut them in these restriction enzymes to cut them. And then what we can do is we can add a DNA fragment to it. And so then every time the bacteria actually copies itself, it also recopies the recombinant plasmid that we want. And so what we're getting it to do is basically, as it replicates, it's getting it to make whatever this piece of DNA we want. All right, let's talk about gel electrophoresis a little bit. We're going to do this in our lab. You're going to see this in the Labster technique. And we used to do this in class where you would actually load a well with DNA, and then you use a power source. So the gel is the one we use is agarose, which is agar red algae product. And it basically has little tiny holes in it, that you can't see. And so what we do is we take a DNA molecule and cut it up with these enzymes. And by doing that, you get different size pieces of DNA. And then you run them, you put a buffer in here, and you run electricity across, and you actually load them on the negative side. And then when you turn it on, this is the positive anode. And so what happens is DNA is negatively charged, and it's negatively charged because of the phosphate groups. So they start heading over, and the smaller pieces actually go faster than the larger pieces. And you end up with these lines, these restriction fragments. Here we have what we call a ladder. These are certain sizes that we already know. So it's kind of like a ruler on the side. So if this one matches with this one, we will know the size of that. The problem is these bands can end up anywhere on the gel, depending on how long you run and how much power you're putting through there. So you need to have some kind of measurement, this ruler on the side. They call it a ladder. All right, and so we're going to be looking at this in lab on how to do a crime scene and also how to do a paternity test. So you will be asked to do that on the quiz for this. All right, and then we have a process I want you to know, PCR, polymerase chain reaction. This is where you're taking a small piece of, you're taking a target sequence of DNA, and what you're trying to do is break it up into smaller little pieces. And then what you do is polymerase is the enzyme that builds DNA. And then what you're going to do is basically add enzymes, and then you're going to also heat to denature this and then cool it back down. And you're going to go through cycle after cycle. And after about 30 cycles, you end up with billions of pieces of DNA. So the whole purpose of PCR was basically to take a small amount of DNA and increase it to make a large amount of DNA. We now use it for crime scenes and things like that. You'll see that in our lab. So we can do this with gene cloning. The same idea is you have a cloning vector and you add that recombinant DNA plasmid into it. So what you then want to do is polymerase chain reaction is you want to get the individuals that have that vector in it to, or that recombinant DNA in it to replicate itself over and over again. And so this is PCR is something you will do if you take Bio8 in the Bio8 lab. All right, and so let's look at expressed clone genes. Bacterial expression, you have an expression vector, which is a highly active bacteria promoter. The problem with trying to clone genes with a bacterial expression is the introns. They're very large, they're hard to manage for the bacteria. Bacteria do not have introns, so it's kind of, what do we do with these things? So it's better to do it in eukaryotes expression. And what they do is to get the clone genes in, they'll actually hit it with pulses of electricity to kind of blow little tiny holes in the nucleus so that the DNA can get in. There are actually now really, really tiny small needles they can poke and push it in. And they're actually now figuring out ways of using bacteria to bring the plasmid in itself. So these are all processes that are lab processes that people are very good at. And there are careers in there if you can do this very effectively. All right, and then there are ways of analyzing gene expression. So this is some of the things they're doing is trying to figure out how things are happening. And so what they're doing is you have in situ hybridization, and what they're doing is they're actually putting markers on different genes and using fluorescence. And then what they can do is follow those genes to see what they do. And here we have the development of a Drosophila larvae. And you can see by the color what gene is actually being physically expressed. So we know a lot about where the genes are and what they're doing to like this embryo because of that color patterns that put out. And actually some of them are quite beautiful. On the right, we have RT-PCR, reverse transcriptase. What happens is it goes DNA to RNA. Reverse transcriptase is to go from mRNA back to DNA. So what we're trying to do there is take the messenger RNA and to rebuild what the DNA would look like. And so reverse transcriptase is also an enzyme you'd find in HIV. That's how HIV is a piece of RNA, and that's how it gets the human body to make its DNA. And then there's RNA sequencing, where you're taking lots of little pieces of RNA and using that reverse transcribed into DNA pieces, and then which is what we talked about here a little bit. And then what you do is you put it all together, map it, and you're looking for overlaps, and it's kind of like a puzzle and trying to get a genome sequence. So all of these tools are being used, and what's happened over time is we're getting faster and faster at it. And we'll talk about that a little bit more when we talk about human genome.